Hello and welcome to today's PhenoRob PhD Graduate Talk. My name is Justus Detring and I'm happy to give you a little lecture today on my PhD thesis titled Establishment of Phenotyping Routines for Assessing Tolerance Levels to Syndrome Bas-Richesse of Different Beta vulgaris Varieties. But before I step into my research, I would like to give you an overview on the pathosystem I mainly work with, which is called the Syndrome de Bas-Richesse. The syndrome de Bas-Richesse is commonly abbreviated with SBR and can be caused by two phytobacteria. Candidatus azonophonus phytopathogenicus, which is a gamma-3 proteobacterium, and Candidatus phytoplasma solani, which is a phytoplasma from the Stolberg group. Both phytobacteria are mainly transmitted by Pentasteridus leperinus, which is a succeed species commonly called reed glass-winged cicada. The disease was firstly reported in Burgundy in France in 1991, where it already caused a tremendous economic loss of 50% on over 1,000 hectares. Since then, SBR spread throughout Central Europe over the last 25 years to countries like Austria, Switzerland, Germany, Romania, Serbia, Slovakia, and so on. Currently, there are not general control measures for SBR, but is partly controllable through crop rotation. And this is because the vector is highly adapted to the common crop rotation sugar beet winter wheat. So by altering winter wheat with a non-host crop to Pentasteridus, this can disrupt the life cycle of Pentasteridus, which then reduces the number of adults in the following year. But unfortunately, due to the high polyphagy of Pentasteridus, it enabled more hosts in the recent years, like carrot and potato, which reduces the efficacy of this method. So tackling SBR on the pathogen side might be more promising. But before I get more into detail here, I would like to give you an overview on the symptoms of SBR in general. So generally, SBR symptoms are highly dependent on the pathogen prevalence. And that means that if the Proteobacterium is more prevalent, we have small, narrow, lancet new shoots, leaf chlorosis, and a brownish discoloration of the taproot tissue. But on the other hand, when the phytoplasma is more prevalent, we have strong canopy wilting and a rubbery taproot comparable to the rubbery taproot disease in Serbia. There's a special feature of the Proteobacterium because it is an insect endosymbiont. And that means that Pentasteridus, the vector itself, is infected with the Proteobacteria. And when it feeds on the phloem sap of sugar beets, it transmits it to the sugar beet, where it then alters the phloem sap concentration by enriching it with sugars and potentially essential amino acids for the insect, which then enhances the sugar beet as a food source for the insect and most likely enabled sugar beet generally as a host for Pentasteridus. So the Proteobacterium is definitely here the game changer in this pathosystem. There are already a few tolerant varieties found to the Proteobacterium, but yeah, more are urgently needed, and especially the traits behind the tolerance need to be described. And here comes my research into handy, which was mainly focusing on establishing methods for assessing the tolerance levels to syndrome de Bas-Richesse and also phenotyping the disease in general. So my PhD was structured into two phases. First phase is method investigation, respectively development, and then applying those methods to experiments with the following data analysis. During those phases, 3 manuscripts were established and published, which structured my thesis and will also structure this lecture today. Here you can see an overview of all the methods which were established and applied for phenotyping syndrome de Bas-Richesse and assess the tolerance levels. And those methods can be generally separated between aiming on the above ground and the below ground organs of sugar beet. But yeah, phenotyping a systemic disease like syndrome de Bas-Richesse is challenging because distinct symptoms like fungal spots are missing, which can be then easily categorized and described. And that's why I chose a highly sensitive method for being my main method for phenotyping SPR, which is called hyperspectral imaging. And hyperspectral imaging is capable of measuring the reflectance properties of objects and also the changes which occur due to different influences, in our case SPR. And that's why it makes it highly valuable for me to be the core method of my PhD. Well, during applying and establishing this method, I noticed that assessing high data quality, in this case, high hyperspectral image quality, is not trivial at all. And that there is no pipeline of assuring or evaluating hyperspectral imaging for plant phenotyping present in the literature. And that's why the goal of my first publication was to establish such a pipeline. But before I go here more into detail, let's step back and talk a little bit about hyperspectral imaging and quality in general. So what is it in general? So when you think of classical RGB images, classical photos from your smartphone, for instance, they are assembled by measuring three key color areas: red, green, and blue. So hyperspectral measurements are capable of measuring way more color areas, and also this in a higher spectral resolutions. We call it, in this case, bands. So more spectral bands are available also in areas which are not visible, like the UV area or infrared, near-infrared area. And this sophisticated spectral measurement can then be used for a detailed analysis. And when we think about hyperspectral image quality, three factors come in mind which influence them the most, which is the sensor in general, which finds the spectral range measuring, and also when we think on hyperspectral imaging, the spatial resolution. Then the illumination, which influences the sensitivity of the measurement, and also the object in general, because measuring a plant canopy, for instance, we have a three-dimensional structure we have to cope with a two-dimensional measurement. Here you can see the pipeline established for assuring the quality of hyperspectral imaging. Due to the lack of time, I would focus on two key aspects in this publication, which is first the illumination, and second, the use case, which was established to check if the quality assured data is really usable or feasible for phytopathological phenotyping use cases. And in this case, we chose Cercospora leaf spot disease as our use case because it is easily categorizable in the spatial dimension. So the spots on the leaves can be labeled easily, and it is then easier to handle data here and really see if the results are feasible for a broader use case. And the quality assurance pipeline and the use case was both tested on a newly developed hyperspectral imaging camera, a mobile camera, which you can see here on the left, which comes with a special feature. And this is the integrated illumination here on the back side, which is a LED illumination. And this LED illumination was tested against the most common illumination for hyperspectral imaging, which are halogen lamps, and against a so-called reference measurement of a spectroradiometer, a non-imaging spectroradiometer. And this test was also done on a sugar beet canopy, as you can see here on the right side. So when we first compare the reference, so-called reference measurement with the halogen lamps, we can see that there is a shift of the curve appearing. Yeah, this is most likely occurring due to the fact that the reference measurement is non-imaging. So here just a small spot is measured on the leaf surface. And with the halogen lamps, we measure the whole canopy. And as you can see here on the right, the canopy is not completely evenly illuminated. But when it comes to radiometric normalization, it is treated as it would be evenly illuminated, which is still the state of the art for plant phenotyping today. When we then have a look on the spectral curve from the LED lamps, we not only see a spectral shift, but also that the course of the curve is changing in some areas, especially in this area here. And when we have a look then on the false color images of this spectral area, we can see that some parts of the canopy are pretty much glowing, and that these areas have reflectance values way above the normal reflectance values for leaves. And this is most likely occurring due to mirroring effects, because the internal illumination, as you saw on the picture, is pointed in a 90-degree angle on the leaves. And when this light then hits the leaf, most likely a waxy surface, yeah, just mirroring effects can occur and then oversaturate the image. So this brought us to the conclusion that, yeah, external illumination of halogen lamps is the best way to assure that the image quality is very high. And that's why we chose also halogen lamps for our use case. As I mentioned before, our use case was to work with Cercospora leaf spot disease. And in this case, we wanted to assess the disease severity. Therefore, a greenhouse trial was established where we infected sugar beets with Cercospora leaf spot and then measured it in a time series event with a hyperspectral imaging system, and then train a machine learning model for segmenting the diseased leaf parts, and also comparing then those results to an expert rating. And the results, you can see them here. The expert rating in the dotted line and the bars resemble the results from the machine learning model prediction. So the trend of both results is the same, even though the expert rating is always a bit higher. But when we have here a look on the false color images, I would say that the model did a pretty good job in segmenting the leaf parts, the diseased leaf parts. So I would assume that the truth is somewhere in the middle, so that the expert a little bit overrated and the model a little bit underrated. But yeah, all in all, we can say that quality assured hyperspectral imaging data is feasible for sophisticated plant phenotyping. Another conclusion from this publication that two-dimensional radiometric normalization on three-dimensional objects can cause spectral shift, and that non-diffuse illumination at a 90-degree angle can cause a strong bias on spectral measurements. With all this knowledge gathered, I was then capable of establishing a sophisticated hyperspectral imaging laboratory setup, which you can see here. In this setup, a hyperspectral imaging line scanner is installed. And due to the fact that it is a line scanner, it is assembled to a linear axis, because line scanner, they record the images line by line. So they need to be moved above the object to then assemble a full image. The setup is illuminated by halogen lamps, 6 of them. And to the halogen lamps, diffusing screens are attached to, evenly illuminate the setup as much as possible and disable mirroring effects. Already at this point of my research, I was curious about phenotyping this disease with more methods beyond hyperspectral imaging. That's why we chose to apply further methods, such as molecular diagnostic, to assess the infection rate in the trials, and also 2D measuring of the leaves, because in literature it is well known that the leaf geometry is affected by SPR. And during my experimental trials, I also noticed that also the taproot geometry is affected. That's why we chose 3D scanning for assessing taproot geometry parameters. Well, at this point, you came probably across the question, why am I focusing so much on laboratory setups? And in fact, I gathered some experience in field trials with SPR, which brought me to the conclusion that when you really want to describe and phenotype a new disease, you have to do it in a closed environment to disable all effects which can disrupt your measurements or influence your measurements other than the desired, in our case, SPR. That's why I established a greenhouse trial where sugar beets were inoculated with the main vector, Pentasteroideus leperinus, and at this point, only carrying the proteobacteria, because at this point in my research, an artificial infection was only possible with the proteobacteria because the phytoplasma in laboratory Pentasteroideus rearings gets lost. And that's why all the results and data analyzation is all focused in this talk today on the proteobacteria. So after inoculation with the insect vector, the sugar beets were then measured in a hyperspectral imaging system, and also the disease incidence was rated. And as I mentioned at the end, invasive methods were applied, such as single leaf 2D scans and 3D scans of the taproot and molecular diagnostic to get a broader picture on this disease. But let's first talk about the hyperspectral imaging data and results. So our goal was to assess the disease incidence from the data so that we can judge by the hyperspectral images whether this sugar beet is infected or not. And as you probably already noticed, hyperspectral imaging data is very vast and complex, and that's why often machine learning methods are applied to support the data analysis. In this case, we used a convolutional neural network, which was fine-tuned to judge whether a hyperspectral image or the sugar beet in the hyperspectral image is infected or not. And in this first shot, we were pretty successful and got a high accuracy of 94%. But as an agricultural scientist, I was very curious about the biological reasons behind it. So why is the network choosing this plant as infected or not? And that's why we applied explainable machine learning, in this case, these heat maps, to see which areas of the picture were the most relevant for the network to make the decision. And in most of the pictures, always the petioles were glowing, which brought us then to the idea that we segment the canopy by its organs, so that we segment the canopy between petioles and leaves. So that's why we fine-tuned another network for segmenting those parts, and then use those masks to focus more on the spectral data. So that's why we trained even more machine learning models, in this case, 2 models based on the spectral petiole data and the leaves data, again with the goal to assess the disease incidence over the pathogenesis. The results are here displayed, and again, we see that the accuracy for the petiole data is way higher for the leaf data. This is probably due to the fact when we have a look at the spectral curve on different time points, that surely the spectral curve of the diseased leaves is shifting from the healthy leaves, but we just see a shift here. So I would assume that the curves or that the curves in general are correlating still pretty much. But when we have a look at the petioles, we see also cause changes in the curve. Even though they are pretty minor, I would say those are distinct features which can be then more easily picked up by a model to make a decision, to learn whether this data is categorized to one group or the other. But when it comes to the spectral regions, which were most important for making a decision, both the petiole and the leaves, the regions between 650 and 660 nanometers are most important. We know from literature that in this region, one of the chlorophyll absorption bands is present. So probably chlorophyll degradation might be here a reason for changing the reflectance properties of either leaves and petioles. As I mentioned, we also applied 2D leaf measurements of single leaves, and there we extracted a lot of parameters, but I only want to display here the most interesting ones. So we know from literature that also leaf biomass reduction is described for SPR, which could be approved by our data, by our results, because leaf area and leaf length is significantly reduced in infected sugar beets. When it comes to leaf symmetry, which is also described in literature, that we have asymmetrical leaves, we couldn't approve this point, so we didn't see a significant change in the leaf symmetry index between non-infected and infected leaves. When we have a look at the results from the 3D scans, we could also approve the first visible changes that, SPR has an effect on the morphology of the sugar beet taproot. So what did we do with the 3D scans? We extracted here also a lot of parameters, and then did a feature selection and feature importance with Boruta, and then with the help of Sharp, we also were able to rank those features in their importance, and also which of the value regions were important for the Boruta Sharp model to make a decision between affected or not. So we can see here, for instance, that low values of biggest notched height versus biggest notched parameter have a higher impact on the model performance than high values. And it's the other way around for roundness fitted biggest circle. Here, high values have a higher impact on the model performance, so are positive for the model performance compared to low values. And when we have a look on the spatial distribution of those features, of a single feature, we see that also the spatial regions where those features are assessed are also important. So for instance, here, the region below the hypocouture is very important, and here we see the region towards the sugar beet taproot is very important. And surprisingly, those regions are also the regions where the pathogen is most prevalent. So those regions are the regions where the morphology is altered the most. And this is, aligning with this, the pathogen prevalence. All in all, we can say that there's a high relevance of sugar beet petioles for SBR disease classification with machine learning based on hyperspectral information. That the most important spectral features for SBR disease classification are between 650 and 606 nanometer, and that SBR affects the morphology of the sugar beet taproot, and that there's a strong variance in the spatial distribution of the taproot morphological alterations. Yeah, due to the very interesting results on the taproot morphology, I was even more curious on research on the taproot, and in this case, a little bit more with a biological background, I would say. So we know that when the pathogen prevalence shifts to the phytoplasma, that rubbery taproots are occurring. So I was curious what happens when it's just a single infection with the Protobacterium. Is the taproot texture changing? And also I was curious on the structural components. Are they then changing as well, aligning to the texture? So that's why we extracted structural components just as such as mark content, alcohol soluble residues like cellulose, lignin, hemicellulose, and pectin. Also the taproot volume, length, and width from the 3D scans to have a little bit more distinct parameters from those scans. Here another greenhouse trial was established. In this case, 3 different varieties, 1 susceptible and 2 tolerant ones. And when we have a look at the data from the 3D scans and the structural components, we see that the susceptible variety, for instance, there's a strong reduction in fresh weight, which is no surprise, an increase in dry matter, and also an increase in mark content and alcohol soluble residues. Also, the volume is strongly increased, which is also decreased, which is also aligning with the literature. When we have a look at the tolerant varieties, we see that there's the decrease in fresh weight is not that much, which was also expected, but we see a stronger increase in alcohol and solvent residues and mark content. And this is aligning with the taproot texture measurements. So here we also extracted a lot of different parameters. Here are displayed the most interesting ones, the most interesting results. So first of all, we measured the periderm firmness, and this is by assessing the puncture resistance of the periderm. And here, this is measured by how much force is needed to penetrate the periderm, measured with this small stick here. And also, the flexural strength was measured. So how much force is needed to break a sugar beet piece apart. So when we have a look at the puncture resistance, we see that the susceptible variety, the infected sugar beets have a significant decreased puncture resistance. And this is also aligning with the observations that we really saw shriveled sugar beets. So here's some sort of, a loss of water, a loss of turgor is happening, which is not as strong for the tolerant varieties. When we have a look at the flexural strength, we see that it is increased for the tolerant varieties. And this is aligning with the increase of the structural components. So somehow here, the varieties, they are, have a little bit stronger tissue compared to the susceptible one. And how does this align with symptoms in general? So the brownish discoloration described for SBR, which is pretty distinct for this disease, is described in literature as lignification. But when we think about lignification, we would then also see an increase in structural components and also probably an increase in tissue strength. So we would assume that the variety, in this case, the susceptible variety with the strongest brownish discoloration has the strongest tissue and also the the higher concentration in structural components, but it's the other way around. We see here less discoloration, but more structural components. So I would assume that this brownish discoloration is probably not lignification, but some sort of occurring due to other defense mechanisms in the plant, stronger defense mechanisms, such a hypersensitive response, which causes, for instance, necrosis. And this is also a little bit aligning with a collaborative research which we have done with the Forschungszentrum Jülich, where we measured the carbon allocation of SBR-infected sugar beets. And those were measured by exposure of the sugar beets to labeled CO2, which then could be tracked by a PET scan to see the carbon flow in the sugar beet, non-infected compared to infected. We see that here the carbon flow in the infected sugar beet is completely disrupted in some areas. And this is also not really what we would expect in a defense mechanism such as lignification. So this defense mechanism in other pathway systems is known to just enhance the cell walls to prevent or to more or less reduce the systemic spread of the disease, but not really completely rupture the vascular system and destroy the plant itself. So this is a little bit more associated with strong defense mechanism like hypersensitive response. So wrapping up the third publication, we can say that SBR causes softening of the sugar beet periderm of susceptible varieties, that increased force is needed to fracture a taproot piece of Proteum bacterium infected tolerant varieties, and that SBR causes a non-significant increase of IER and mark content in sugar beet. But really further investigations are needed with more varieties and more repetitions to verify those results. Here, once again, the overview of all the methods which are applied in my PhD for phenotyping SBR. And at this point, I would really like to wrap up my PhD and move on to the thesis summary and key findings. Well, the illumination property and angles strongly influence hyperspectral image quality. Canopy organ segmentation can enhance phenotyping results. As of Proteobacterium-induced alteration in canopy reflectance between 650 and 660 nanometer, especially in the petioles, are present. SBR disease incidence can be estimated by machine learning models based on canopy reflectance properties during pathogenesis with high accuracy. And the Proteobacterium-induced taproot morphological alterations can be parameterized with 3D scanning and texture analysis. And in general, this characterization provides a foundation for the assessment of SBR tolerance levels across different sugar beet varieties. But in the end, more research is needed. So I would like to present you here further challenges and future perspectives on my research topic. So I would further integrate the third dimension into radiometric normalization of hyperspectral images for plant phenotyping. It is very important to generalize the spectral morphological characterization of SBR by testing more sugar beet varieties. Single leaf and petiole analysis can be a possible base for SBR disease severity estimation. And definitely the robustness of the collected parameters need to be investigated under uncontrolled conditions. And to understand more the defense mechanisms of the sugar beet, I would suggest doing fiber analysis, single fiber analysis, and for instance, a depth staining to check for the reactive oxygen species caused by a hypersensitive response. At this point, I'm at the end of my talk, and if you have any questions, just reach out to me my email address here displayed. Yeah, then thank you very much for this opportunity to present my research in this talk series. And yeah, see you around. Bye-bye.